Hi, I'm going to present you a quantitative eDNA-based method to monitor spawning activity of two emblematic fish species in Lake Geneva. Perch and whitefish are the two main species for Lake Geneva fisheries. Anthropogenic pressures and climatic change have an impact on their captures. And to follow population fluctuations, a monitoring of their spawning activity is performed each year, but unfortunately, using destructive methods. The aim of our study is to develop a non-invasive and quantitative method, based on eDNA, to monitor fish spawning activity in lake. In parallel to traditional monitoring, several 2-liter subsurface water samples were collected each week, during the two different spawning periods. Water was filtered through sterile cartridges, and after DNA extraction, samples were analyzed using droplet digital PCR, targeting the Chloé mitochondrial gene of both species in parallel. Firstly, we observed an important eDNA signal variability between the samples collected within a same spawning area, and then, we chose to pool together the discrete samples for each sampling date. We compared those results to the traditional monitoring data, and observed a good match between the two, with the same spawning timing, duration and peak, for both species. We also showed that 50% of the counts are reached at close dates, confirming that the eDNA approach provides similar information to traditional monitoring. Finally, for the non-spawning species, the eDNA signal does not show any variation, and acts as a biological control, which confirms that the eDNA signal variations observed are related to spawning activity. In this study, we showed for the first time in a lake, the efficiency of eDNA associated with ddPCR, to identify the timing, the duration and the peak of the spawning period of two fish species. And that we reached an operational level to implement this non-invasive monitoring in lakes. We also raised the need to include a biological control, which consists to monitor eDNA signal of a fish species that does not spawn during the studied period. Finally, we observed a variability of the eDNA signal in samples collected within the same spawning area. And then, the need to perform multiple water samples in the same area, in order not to miss the spawning eDNA signal. Thank you for your attention. And don't hesitate if you have any question.